The study of other animals, especially a mammal like a mouse, has already allowed us to understand human biology in so much greater detail. That being said, humans are not mice, and not everything that is learned from experiments with mice can be applied to humans, or at least to varying degrees. And so if one were to ask, what is the effect of hormones and hormone receptors on gender-specific behavior and sexual preferences in mice? This may be of interest with some application potentially to humans, and it may not. In other videos, I go into the steroid hormones and the receptors in far greater detail. Just some of the complexity before I discuss mice is that estrogen is made from testosterone. So women make both estrogen and testosterone. Estrogen binds to one of two estrogen receptors, estrogen receptor alpha or beta, and then together they enter the nucleus and attach to DNA and turn on the expression of genes. Testosterone inside cells is converted either to dihydrotestosterone or to estrogen. And so males then require both the androgen receptor and estrogen receptors for masculinization. And just as the estrogen receptor, androgen receptors bind their hormones and then attach to DNA and change the expression of genes. In mice, there are behaviors that males typically display, such as mounting, thrusting, ejaculation, and behaviors that females typically display, such as lordosis, the curvature of the spine during mounting. Mice can also display preferences for a mating partner, whether it be an actual mouse or the odors of male or female mice. Experiments known as gene knockout experiments can eliminate the function of one gene and one gene only, and then when one observes the difference in, say, a mouse, one can determine what was the function of that gene. Expression of estrogen receptor beta is required for the female behavior of lordosis. Male behaviors require the expression of both the androgen receptor and estrogen receptor alpha. This makes sense since that testosterone inside cells can be converted into one of two active forms, dihydrotestosterone and estrogen. Androgen receptors are required for the male odor preferences in which they prefer females. If females are exposed to androgen, for example, if androgen is applied to the females after birth, the female behaviors persist, but they display more male behaviors and they have an increase in aggression. They have an increased interest in female odors. If androgen receptors are overexpressed in males, they have an interest in both male and female partners, and male behaviors are retained and can be exaggerated. If males are exposed to additional androgen, one observes the same results as when excess androgen receptor is expressed. If males are exposed to low levels of androgen or no androgen, they display fewer male behaviors and more female behavior, and also have an increased interest in male odors. The same result is observed if males lack androgen receptor, where they display more female behavior and have an interest in male odors. And thus, there are two ways that male mice may increase interest in male partners. One is that they have excess exposure to androgen, and one is that they are exposed to too little. There is far more to the study of reproductive behavior and gender in mice. However, this points out what was expected, that hormones and receptors for hormones influence these processes. What determines human sexual attraction and many aspects of human gender, this is not yet fully known. However, it is known, for example, that if females are exposed to excess androgen, and there are a number of ways that this can happen, for example, in congenital adrenal hyperplasia, and this and others I review elsewhere, it is known that this has diverse effects, including increasing the likelihood that women will describe themselves as being more attracted to other women. Hormones and their receptors seem to influence many aspects of gender and attraction in both humans and mice.